The purpose of meiosis. Said short, the purpose of meiosis is to produce sex cells through various separations. Meiosis also reduces the number of chromosomes to keep the chromosome count consistent across generations. Meiosis 1. Meiosis 1 begins with prophase 1, during which chromatin condenses into chromosomes. The centrals separate and the spindles begin to form. As this happens, the nuclear envelope begins to break down. Moving on to metaphase. In metaphase, crossing over occurs where genetic material is switched between pairs of chromosomes. Following this, the pairs of chromosomes, which are now crossed over, begin to align at the center of the cell, and is now connected to the centromes by a spindle fiber. The pairs of chromosomes separate, pulled towards the poles of the cell. At this time, the chromosomes are gathered together and a new nuclear envelope forms, which is the process of telophase 1. As the cells separate between telophase 1, they enter interkinesis, at which point two daughter cells are formed and split. At this point, the cell contains N, meaning that they each have 23 double chromosomes. Meiosis 2. In meiosis 2, essentially the same process occurs, except that the process does not involve DNA replication. The process of meiosis together produces 4 cells all told. Each cell now contains 23 single chromosomes. 2N versus N cells. 2N cells, also called diploid, have twice as many chromosomes as N cells, also referred to as haploid. In humans, the diploid number is 46, which is divided into 23 between each daughter gamete. After fertilization, each zygote becomes diploid. Non-disjunction. Non-disjunction occurs as an error of meiosis 1 or 2. Non-disjunction leaves one daughter cell empty without chromatid, while the other receives an extra copy of this chromatid. Non-disjunction is the cause of Down syndrome, Edwards syndrome, and Patou syndrome. Also, it results in an infertile sex cell. The variety in gametes. Sexually reproducing organisms have an advantage over asexual organisms because of the resulting variation in the offspring. This is achieved with a gamete through the process of crossing over. This is where the chromosomes exchange corresponding sections of the DNA with other chromosomes. Diversity of the gametes is also achieved through independent assortment. Each individual chromosome is selected randomly to be put into the gamete. This produces a greater number of possibilities for a gamete's possible genetic codes. More variety is achieved when random gametes fertilize each other.